Find somewhere comfortable. You don't need to sit up straight or hold anything in your mind. You don't need to follow closely or remember any of this or do anything at all except let the words arrive and drift past the way sounds do when you're almost asleep. If today was hard, and for many people today was hard, you can set it down now. Not forever, just for tonight. The day has already happened. It lives behind you now, growing quieter with every breath. Tonight we're going somewhere small, somewhere most people have never really thought about, even though it is happening inside them right now, in this very moment, in every cell of your body, simultaneously without pause. We're going inside the mitochondrion, an ancient thing, a patient thing, something that has been here in one form or another for nearly 2 billion years. You don't need to picture it precisely. You don't need to concentrate. Just let the words come to you, the way warmth does when you pull a blanket close. We're in no hurry tonight. There is something living inside you that is not quite you. It has its own DNA, its own membranes, its own ancient lineage, stretching back through time to a world so different from ours that it is almost impossible to picture. A world without animals, without plants, without anything you would recognize as visible life. A world of slow chemical gradients and warm, shallow seas and microscopic things drifting in the dark. It is the mitochondrion, and the story of how it came to be inside every one of your cells is one of the most quietly astonishing stories in all of biology. To understand it, you have to go back, a very long way back. Somewhere around 2 billion years ago, the Earth was a profoundly different place. The atmosphere was only just beginning to fill with oxygen, a gas that, at the time, was something close to a poison for most of the life that existed. The dominant organisms were bacteria, tiny, single-celled, enclosed by a single membrane, carrying their genetic material loose inside them rather than tucked away in a nucleus. Among them were two very different kinds of cell. One was a larger, more complex microbe, what biologists call an archaeon, from the Greek word for ancient, a cell that had developed a nucleus to house its DNA, that had grown more elaborate over time, but that still struggled to do much with the oxygen accumulating around it. The other was a smaller bacterium, a compact and nimble thing that had evolved a remarkable trick. It could take oxygen and use it to extract enormous amounts of energy from organic molecules. It was, in effect, an engine. What happened next was not a war. It was not a conquest. It was something stranger and more intimate than either of those things. One cell engulfed the other. This happens all the time in the microbial world. Larger cells swallow smaller ones as a matter of routine, digesting them for nutrients. But on this particular occasion, in this particular encounter, and we do not know exactly where or when, only that it happened, the smaller cell was not digested, and the two of them, slowly, over immense stretches of time, became something neither had been before. They became a partnership, and from that partnership, eventually, came every complex cell on Earth, every animal, every plant, every fungus. This idea, that the mitochondrion is a descended remnant of that ancient bacterial loggia, is called the endosymbiotic theory. The word endosymbiotic comes from the Greek, endo meaning within, and symbiosis meaning living together. Living within, together. And for most of the 20th century, the scientists who proposed it were largely dismissed. One of the earliest was a French biologist named Paul Portier. In 1918, he published a book arguing that the structures inside cells that would later be called mitochondria were not simply parts of the cell, but were living entities in their own right, ancient microbes that had taken up residence inside a host and never left. The scientific establishment of the time found this idea eccentric, even offensive. Portier was not taken seriously. His work was set aside. For decades, the question lay dormant. Then, in 1963, two scientists named Margit Nass and Sylvan Nass, working at the Wenner Gren Institute in Stockholm, made a discovery that would eventually change everything. They were looking at mitochondria under an electron microscope when they noticed something that should not have been there: threads of DNA. Not the long, linear chromosomes of the cell's nucleus, but small, circular loops, exactly the kind of DNA found in bacteria. Mitochondria, it turned out, had their own genetic material, their own blueprint, separate from the rest of the cell, written in the same ancient script as bacterial genomes. This was a remarkable finding, but it was still not enough to overturn the consensus. That would take Lynn Margulis. Margulis was an American biologist, and in 1967 she submitted a paper to a series of scientific journals. It was rejected by roughly 15 of them before finally being published in the Journal of Theoretical Biology. The paper was called On the Origin of Mitosing Cells, and in it she laid out, in careful and systematic detail, the evidence that the mitochondrion was not something the cell had invented from scratch, but something it had acquired, something it had once swallowed and kept. The evidence she marshaled was, in retrospect, overwhelming. There was the DNA, that small circular molecule, so unlike anything in the cell's nucleus, so unmistakably bacterial in its form. There were the ribosomes, the tiny molecular machines that read genetic instructions and build proteins. The ribosomes inside a mitochondrion are not like the ribosomes floating in the rest of the cell. They are smaller, and structurally they match the ribosomes of bacteria far more closely than they match the ribosomes of the host cell's own cytoplasm. Antibiotics that specifically target bacterial ribosomes, drugs like chloramphenicol, also disrupt mitochondrial ribosomes while leaving the cell's other ribosomes entirely unaffected. This is not a coincidence. And then there was the membrane, or rather, the membranes, because the mitochondrion is wrapped in not one, but two. And that double wrapping is itself a fingerprint of the original event. The outer membrane is thought to be derived from the host cell's own membrane, the remnant of the moment of engulfment, while the inner membrane is the original bacterial membrane of the ancient loggia. Ancient, preserved, still doing what it was doing 2 billion years ago. Margulis's paper was initially met with resistance. The idea that something so fundamental about the architecture of life had arisen from an act of cellular hospitality or cellular accident was difficult for many to accept. But the evidence accumulated, genome sequencing, which became possible decades later, confirmed it beyond any reasonable doubt. The mitochondrion's DNA is most closely related to a group of bacteria called alpha proteobacteria. Its closest living relatives are free-living bacteria that still exist today, swimming in lakes and soil and ocean water. The ancient ancestor of every mitochondrion was once, unambiguously, its own creature. It is worth sitting with that for a moment. Inside every cell of your body. And then, 2 billion years ago, one of their ancestors was swallowed by something larger, and instead of being destroyed, it adapted. It gave up its freedom piece by piece, transferring most of its genes to the host cell's nucleus over evolutionary time, becoming more and more dependent, more and more integrated, until the boundary between guest and host became almost impossible to locate. Almost, but not entirely. Because the mitochondrion still carries its own DNA, still replicates on its own schedule, still has membranes that speak of its bacterial past. It is not quite you, and it is not quite not you. It is something in between, an ancient stranger that has been living inside you, and inside every ancestor you have ever had, for longer than animals have existed on this planet. The number of mitochondria in a single human cell varies enormously depending on what that cell does. A red blood cell, which is stripped of almost everything in order to carry as much oxygen as possible, has none at all. But a liver cell, which has enormous metabolic demands, may contain somewhere between 1,000 and 2,000. A heart muscle cell beating without pause every second of your life may hold several thousand, and each one of those is a descendant of that original bacterial guest, still humming, still working, still ancient. There is something quietly moving about that, something that feels, in the dark and the quiet of wherever you are right now, worth holding gently for a moment before we go further in. If you could shrink yourself small enough, past the scale of cells, past the scale of bacteria, down into the scale of the very large molecules, and approach a mitochondrion, the first thing you would encounter would be its outer surface. It looks, at this scale, like a smooth oval, something like a very small bean or a slightly flattened egg. Its outer boundary is the outer mitochondrial membrane, and it is, relative to what lies within, a fairly open and permeable thing. Embedded throughout it are proteins called porins, channel-forming proteins that act like open doorways, allowing small molecules and ions to pass through relatively freely. Water moves through, nutrients move through, signaling molecules move through. The outer membrane is not trying to keep the world out. It is a boundary, but a gentle one. Step through it, and you find yourself in a narrow corridor. This is the intermembrane space, the gap between the outer membrane and the inner one. It is not empty. It is crowded with proteins, many of them deeply important. Among them, resting quietly in the ordinary course of things, is a protein called cytochrome C, which plays a role in energy production and, under very different circumstances, in something far more consequential. We will come back to that. But now, ahead of you, is the inner membrane. And it is extraordinary. The inner mitochondrial membrane is not smooth. It is folded repeatedly and deeply into structures called cristae, ridges and shelves and tubular projections that extend inward into the mitochondrion's interior. The word cristae comes from the Latin for crests, and that is what they are, crests, folds, a vast and complicated terrain of membrane that, if you were to unfold it and lay it flat, would cover an area many times greater than the outer membrane surrounding it. In a cell with very high energy demands, a heart muscle cell, for instance, the cristae are packed so densely that they fill almost the entire interior space. This is deliberate. The inner membrane is where the most important work of the mitochondrion happens, and surface area is everything. More membrane means more space for the molecular machinery of energy production. The cristae are the solution to a fundamental engineering problem. How do you fit an enormous working surface into a very small space? You fold it. You fold it again. You keep folding. But the inner membrane is more than just folded. It is also, by design, almost completely impermeable. This might sound like a flaw. In fact, it is the whole point. The inner membrane is one of the most selectively impermeable biological membranes known. Almost nothing crosses it without being specifically escorted by a dedicated transport protein. Protons, hydrogen ions, individual hydrogen atoms stripped of their electrons, cannot cross it freely. This impermeability is essential to how the mitochondrion generates energy, and we will follow that mechanism in detail in the next chapter. For now, the key thing to understand is that the barrier is intentional. The inner membrane is a dam. Its impermeability is what makes it possible to store potential energy in the form of a gradient, a difference in concentration, across it. Part of what gives the inner membrane its unusual character is a molecule called cardiolipin. Cardiolipin is a phospholipid, a type of fat molecule that forms the basic fabric of all biological membranes, but it has an unusual structure. Most phospholipids have 2 fatty acid tails. Cardiolipin has 4, arranged around a central glycerol backbone, giving it a shape and a set of physical properties that are quite distinct from the phospholipids found in most other membranes. It is particularly good at maintaining the tight curvature of the cristae folds, and it helps stabilize the protein complexes embedded in the inner membrane that carry out the work of energy production. Cardiolipin is also found in bacterial membranes. This is one of those quiet details that stops you if you think about it carefully. It remains like an old handwriting style that persists through generations, a faint but legible signature of where the mitochondrion came from. Pass through the inner membrane and you arrive at the innermost space, the matrix. The matrix is the fluid-filled interior of the mitochondrion, and it is dense with activity. It contains the enzymes responsible for the central metabolic cycle that extracts energy from nutrients. It contains the proteins involved in building and copying mitochondrial DNA. And it contains the DNA itself. Mitochondrial DNA is a small circular molecule. In humans, it encodes 37 genes, a tiny number compared to the roughly 20,000 genes in the cell's nuclear DNA. Most of the genes that the ancestral bacterium once carried have, over evolutionary time, been transferred to the nucleus, where they are now maintained and expressed by the host cell's own machinery. What remains in the mitochondrion's own genome is a compact, essential set, 13 genes encoding proteins that are core components of the energy-producing machinery, and 24 genes encoding the RNA molecules needed to read and act on those instructions. The circular shape of this DNA is significant. Linear chromosomes, like the ones in your cell's nucleus, require special caps at their ends, structures called telomeres, to protect them from degradation. Circular DNA has no ends. It is, in this sense, more elegant, more ancient. Bacteria carry circular DNA. The mitochondrion carries circular DNA. The mitochondrion carries circular DNA. Scattered through the matrix alongside the DNA are the mitochondrion's own ribosomes, those molecular machines that build proteins from genetic instructions. As noted before, these ribosomes are structurally closer to bacterial ribosomes than to the ribosomes elsewhere in the cell. They are, in a sense, the mitochondrion's own voice, a way of reading its own genetic instructions and building its own proteins, independently of the cell's central machinery. It is worth pausing here to appreciate what this architecture means in terms of scale. A typical mitochondrion is somewhere between about 3/4 of a millimeter and 3 millimeters across. No, wait, let me give you the right sense of this. A millimeter is 1,000th of a meter. A mitochondrion is measured in micrometers, millionths of a meter. So a typical mitochondrion is between roughly 3/4 and 3 micrometers in its longest dimension. A human hair is about 70 micrometers wide. You could line up somewhere between 20 and 100 mitochondria across the width of a single hair. And yet inside that space there is an outer membrane, an intermembrane space, an inner membrane folded into elaborate ridges, a matrix filled with enzymes and DNA and ribosomes, and the entire molecular machinery of one of the most sophisticated energy conversion systems ever produced by evolution. It is a room within a room within a room, a set of nested spaces, each one more intimate than the last, each one playing a specific role in a process that has been running, essentially unchanged in its fundamental logic, for nearly 2 billion years. Wherever you are right now, in a bed, on a sofa, somewhere quiet, your cells are full of these nested rooms, thousands of them perhaps in each cell, each one humming with its own quiet activity, ancient and patient and deeply, deeply familiar with the work. Every movement you have ever made, every thought you have ever had, every word you have ever spoken, all of it was paid for by a molecule called ATP. Adenosine triphosphate. The name is less important than the thing itself. ATP is the cell's universal energy currency. When a muscle fiber contracts, it spends ATP. When a neuron fires, it spends ATP. When a cell builds a new protein or repairs its DNA or pumps ions across its membrane to maintain its electrical charge, ATP, always ATP. The cell does almost nothing without it. An adult human body generates and consumes something close to its own body weight in ATP every single day, not a reserve that sits somewhere and is slowly drawn down. ATP is made and spent and remade continuously in an extraordinary cycling of the same molecule through billions of transactions per second. And the place where the vast majority of that ATP is made is the inner membrane of the mitochondrion. To understand how, we need to follow a molecule of glucose, the sugar that arrives in your bloodstream after a meal, from the moment it enters the cell all the way down to the spinning of a molecular motor. The journey begins not in the mitochondrion but in the cytoplasm, the fluid that fills the cell outside the organelles. Here, glucose, a 6-carbon molecule, is broken apart through a sequence of 10 chemical reactions collectively called glycolysis. The word means, roughly, sugar splitting, and that is what it does. By the end of glycolysis, the 6-carbon glucose has been divided into 2 3-carbon molecules called pyruvate. Along the way, a small amount of ATP is produced, a net gain of 2 molecules of ATP per glucose, and 2 molecules of a carrier called NADH are generated. NADH is worth understanding properly because it is central to everything that follows. NADH stands for nicotinamide, adenine dinucleotide in its reduced form, but what matters is not the name but what it does. Think of NADH as a delivery vehicle for high-energy electrons. When glucose is broken apart, energy is released. Some of that energy is captured immediately as ATP, but much of it is captured instead by loading it onto electron carriers like NADH. The NADH molecule picks up a pair of high-energy electrons, along with a proton, and carries them to the mitochondrion, where they will be used to drive the main energy-generating machinery. It is not the final product, it is the means by which energy is transported from one place to another. A similar carrier, called FADH2, works alongside NADH. It carries electrons at a slightly lower energy level for reasons that will become relevant shortly. Now, the pyruvate molecules, produced by glycolysis, move from the cytoplasm into the mitochondrial matrix. They pass through specialized transport proteins in both membranes. The outer membrane is relatively open, but the inner membrane requires a dedicated carrier, and once inside the matrix, each pyruvate molecule is transformed. An enzyme complex strips off one of the 3 carbons, releasing it as carbon dioxide, which is why you breathe out carbon dioxide. It is a waste product of this process, and what remains is a 2-carbon fragment called an acetyl group. Another NADH molecule is generated in the process. Acetyl-CoA is the entry point for the next stage, the citric acid cycle. The citric acid cycle is a loop of 8 chemical reactions that takes place entirely within the mitochondrial matrix. The 2-carbon acetyl group from acetyl-CoA is fed into the cycle by being attached to a 4-carbon molecule already present, producing a 6-carbon compound. The cycle then progressively oxidizes this compound, meaning it strips electrons from it, releasing both carbons as carbon dioxide, regenerating the original 4-carbon molecule ready to accept the next acetyl group, and in the process producing a rich harvest of electron carriers, 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of FADH2, and 1 molecule of FADH2, and 1 molecule of ATP. Since 1 glucose produces 2 acetyl-CoA molecules, the cycle turns twice per glucose. So far, the total ATP yield from glycolysis and the citric acid cycle is relatively modest, around 4 molecules of ATP per glucose. But the real harvest is in the electron carriers, 10 NADH molecules and 2 FADH2 molecules, all of them laden with high-energy electrons, all of them now converging on the inner mitochondrial membrane. This is where the process becomes genuinely extraordinary. The inner membrane contains 4 large protein complexes, enormous molecular machines embedded in the membrane's fabric, each one a different size and shape, each performing a specific step in a relay. They are simply called Complex 1, Complex 2, Complex 3, and Complex 3, and together they form what is called the electron transport chain. NADH approaches Complex I and donates its pair of high-energy electrons. Complex I accepts them and passes them along its internal structure, a chain of ion-containing clusters that hand the electrons from one to the next, like a relay race, each handover releasing a small amount of energy. That energy is not wasted. It is used immediately to pump protons, hydrogen ions, from the matrix through the complex and into the intermembrane space. 4 protons are pumped for every pair of electrons that passes through Complex 1. FADH2 donates its electrons not to Complex I but to Complex 2, which does not pump protons. This is why FADH2 generates slightly less ATP in the end than NADH does. The electrons from Complex 2 join the relay at a later point. The electrons now passed from Complex 1 or 2 are picked up by a small mobile molecule called ubiquinone, sometimes called coenzyme Q, which diffuses through the inner membrane carrying the electrons to Complex 3. Complex 3 accepts them, passes them through its own internal relay, and in doing so pumps 4 more protons into the internal relay, and in doing so pumps 4 more protons into the intermembrane space. The electrons are then carried to Complex 4 by another mobile carrier. This one, a small protein called cytochrome C, the same molecule that sits quietly in the intermembrane space and has a very different role to play under conditions of cellular stress. Complex IV receives the electrons from cytochrome C and uses them to perform the final act of the chain, combining electrons with protons and with oxygen to produce water. This is why oxygen is necessary for aerobic respiration. It is the final electron acceptor, the destination at the end of the relay. Without oxygen, the electrons have nowhere to go. The relay stalls and the entire chain stops. 4 more protons are pumped into the intermembrane space as Complex IV does its work. Now step back and look at what has happened. Through the activity of complexes I, 3, and IV, protons have been pumped from the matrix into the intermembrane space. The intermembrane space is becoming crowded with protons. The matrix is becoming depleted. A gradient has been established, a difference in both concentration and electrical charge across the inner membrane because protons carry a positive charge and their accumulation on one side makes that side electrically positive relative to the other. This gradient, this stalled difference, is called the proton motive force. The name captures it well. It is a force, a potential, like water held behind a dam or a compressed spring. The protons on the intermembrane side are under pressure to flow back into the matrix, but the inner membrane's impermeability means they cannot simply diffuse through. There is only one place they can go: through ATP synthase. ATP synthase is the 5th complex of the inner membrane, and it is unlike the others. It is not simply a pump or a relay. It is a genuine rotary motor, a machine that spins. It has 2 main parts: a portion embedded in the membrane through which protons flow, and a portion that projects into the matrix where the actual synthesis of ATP takes place. As protons pour through the membrane-embedded portion, they cause it to rotate. The rotation is transmitted to the matrix portion, which spins inside a fixed surrounding structure. This mechanical rotation, at rates of hundreds of revolutions per second under optimal conditions, drives a series of conformational changes, meaning physical shape changes in the matrix portion that force ADP and inorganic phosphate together, bonding them into ATP. This mechanism, the coupling of a proton gradient to the mechanical rotation of a molecular motor to produce ATP, was proposed by a British chemist named Peter Mitchell in 1961 at a time when most biochemists expected energy production to involve some kind of direct chemical intermediate. Mitchell's idea, which he called the chemiosmotic hypothesis, chemiosmotic meaning relating to the movement of ions across a membrane, was initially dismissed as eccentric. He was awarded the Nobel Prize in Chemistry in 1978. The idea was not eccentric, it was correct. The total yield of ATP from one molecule of glucose, following the full pathway through glycolysis, the citric acid cycle, and the electron transport chain, is somewhere around 30 to 32 molecules of ATP. Some older textbooks give the number as 36 or 38. The difference reflects updated measurements of how many protons are needed per ATP and how much proton leakage occurs across real membranes. The precise number varies slightly between cell types and conditions, but the order of magnitude is not in doubt. Oxidative phosphorylation, the coupling of the electron transport chain to ATP synthase, is responsible for the overwhelming majority of the energy a cell produces. And it runs on a spinning motor, a molecular turbine turning hundreds of times a second inside a structure smaller than a bacterium. Inside a cell so small you cannot see it without a microscope, inside a body that is doing this in trillions of cells simultaneously, right now while you rest. The universe has produced many extraordinary things. This is one of them. When most people picture a mitochondrion, they picture something static, a fixed, bean-shaped structure sitting in the cell like a piece of furniture, a stable organelle doing its work in one place. This picture is almost entirely wrong. Mitochondria are among the most dynamic structures in the cell. They move, they change shape constantly, they extend long arms reaching through the cytoplasm. They merge with one another, two separate organelles flowing together into a single, larger, connected structure. This continuous process of merging and splitting is called mitochondrial dynamics, and it turns out to be deeply important for the health of the cell, the quality of the mitochondria themselves, and, as we will come to, for something as intimate as how tired you feel right now. The merging process is called fusion. When two mitochondria fuse, their outer membranes join first, and then their inner membranes join, and eventually their matrices blend together, mixing their contents, their DNA, their enzymes, their electron carriers, into a shared pool. This mixing is significant. It means that if one mitochondrion has accumulated some damage, a mutation in its DNA, or a protein complex that is not working properly, that damage can be diluted by the healthy contents of its fusion partner. Fusion is, among other things, a quality control mechanism, a way of averaging out imperfections across a network. The proteins responsible for fusion are called mitofusins, specifically mitofusin 1 and mitofusin 2, which sit on the outer membrane and bring adjacent outer membranes together, and a protein called OPA1, which sits on the inner membrane and drives the fusion of the inner membranes once the outer membranes have joined. These proteins work by undergoing shape changes that physically draw the membranes into contact and then catalyze the merging of the lipid bilayers. It is a mechanical process driven by the energy stored in the proteins themselves. The splitting process is called fission, and it is driven by a different set of proteins. The central player is a protein called DARP1, short for dynamine-related protein 1, which normally floats in the cytoplasm in an inactive form. When a mitochondrion is marked for division, DARP1 is recruited to the outer membrane by a set of receptor proteins, including one called MF, short for mitochondrial fission factor, where it assembles into a ring around the mitochondrion at the site where the division will occur. The ring tightens like a purse string, constricting the mitochondrion at that point. The constriction is often initiated at sites where the mitochondrion has made contact with another organelle called the endoplasmic reticulum, a network of membranes elsewhere in the cell, which appears to mark the division site before DOCTAT1 arrives. Once the DARPA wing has tightened sufficiently, the two daughter mitochondria pinch apart. What determines whether a mitochondrion fuses or divides at any given moment? The answer is not simple, but a few principles are clear. A cell that is healthy, well-nourished, and not under particular stress tends to maintain its mitochondria in long, interconnected networks, a kind of mitochondrial reticulum branching through the cytoplasm. These networks are efficient. They allow the rapid redistribution of energy across the cell, the sharing of resources between regions, and the buffering of local fluctuations in ATP demand. They are, in a sense, the cellular equivalent of a well-maintained power grid, interconnected, resilient, able to reroute when one section is under strain. A cell under stress, starved of nutrients, exposed to toxins, dealing with an infection, or simply exhausted, shifts toward fragmentation. The fission rate increases relative to the fusion rate, and the long networks break apart into smaller, more isolated units. This shift is not random. It serves a purpose. When a mitochondrion is damaged, when its membrane potential drops, when its DNA accumulates mutations, when its protein complexes begin to malfunction, it needs to be removed. The cell has a dedicated system for this, a process called mitophagy, which is a specific form of autophagy. Autophagy, from the Greek for self-eating, is the cell's general mechanism for breaking down and recycling its own components. Mitophagy is autophagy, targeted specifically at mitochondria. The process works roughly like this. A damaged mitochondrion which has lost its membrane potential, the electrical gradient across the inner membrane, accumulates a protein called PNK1 on its outer surface. Normally PNK1 is rapidly imported into the mitochondrion and degraded so it doesn't build up. But when the membrane potential is gone, the import machinery stops working, and PNK1 accumulates on the outer membrane. PNK1 then recruits another protein called parkin, which tags the mitochondrion's outer membrane proteins with molecular labels called ubiquitin chains. These labels are recognized by the cell's autophagy machinery, which engulfs the damaged mitochondrion in a membrane sac and delivers it to a cellular compartment called the lysosome, where it is broken down and its components recycled. The connection to fission is essential. For mitophagy to work, the damaged section of a mitochondrion needs to be physically separated from the rest of the network. If mitochondria remained permanently fused into one continuous structure, a localized region of damage could not be isolated and removed without destroying the entire network. Fission allows the cell to cut away the damaged portion, to isolate the failing section, before it can spread its dysfunction to neighboring mitochondria. This is the image of the city's power grid, and it is a good one. When a substation fails, the sensible response is not to leave it connected to the rest of the network and let the fault propagate. Fission is the disconnection. Mitophagy is the repair crew. The balance between fusion and fission is also, it turns out, sensitive to the sleep-wake cycle in ways that researchers have only recently begun to understand. During wakefulness, particularly prolonged wakefulness, the balance in certain neurons shifts toward fragmentation. During sleep, particularly the deepest stages of sleep, it shifts back toward fusion and interconnection. The network reforms. The damaged sections have been cleared, the grid is restored. This is not a metaphor. It is happening in your neurons right now as you rest. The proteins are at work. The rings are loosening. The membranes are flowing together. The cell knows what sleep is for, even if it cannot tell you. There is a process happening somewhere in your body right now that is keeping you alive. Not the heartbeat, not the breath, something quieter than either of those. Cells are dying, deliberately, carefully, and at precisely the right moment. This is not damage, it is maintenance. The process is called apoptosis, a word from the Greek describing the falling of leaves from a tree in autumn. It is programmed cell death, a set of molecular instructions that, when activated, cause a cell to dismantle itself from within in an orderly, contained, and remarkably precise way. No leakage of cellular contents into surrounding tissue, no inflammation, no collateral damage, just a cell reaching the end of its useful life or finding itself beyond repair and carrying out a sequence of steps that results in its own quiet dissolution. Apoptosis is happening in your body at a rate of somewhere between 50 billion and 70 billion cells per day. Most of those cells are in your gut lining, your immune system, your skin, tissues that turn over rapidly, replacing old cells with new ones in a continuous cycle. But the process also removes cells that have accumulated too much DNA damage, cells that have been infected by a virus, cells that are showing the early signs of becoming cancerous. Apoptosis is, among other things, one of the body's most important defenses against cancer. It is the mechanism by which a damaged cell can choose, if that word can be used for a molecular process, to remove itself before it causes harm. And the mitochondrion is at the center of it. The pathway we are interested in is called the intrinsic pathway of apoptosis. Intrinsic because it is triggered from within the cell by the cell's own assessment of its condition rather than by signals from outside. It begins with stress. The cell may have sustained severe DNA damage from radiation or chemical exposure. Its protein folding machinery may have become overwhelmed. It may have detected that its DNA replication is irreparably faulty. Whatever the trigger, the result is a shift in the balance of proteins that regulate the mitochondrion's outer membrane. The outer membrane is normally kept stable by a family of proteins called the BCL2 family, named after the gene in which the first member was discovered. Within this family, some proteins promote survival by keeping the outer membrane intact. Others, when activated by cellular stress signals, act to destabilize it. When the pro-death members of this family gain the upper hand, they gather on the outer membrane and form structures, channels, essentially, that make the membrane selectively permeable in a new and consequential way. This is called mitochondrial outer membrane permeabilization. The name is precise. The outer membrane becomes permeable, and through the gaps that form, something flows out. Cytochrome C. You have already met cytochrome C in the context of the electron transport chain, where it serves as a mobile electron carrier between complex 3 and complex IV, a small, soluble protein that ferries electrons through the intermembrane space as part of the normal business of energy production. Under normal circumstances, cytochrome C stays in the intermembrane space, doing its job, entirely unremarkable. But when the outer membrane is permeabilized and cytochrome C escapes into the cytoplasm, the general interior of the cell, it becomes something entirely different. It becomes a signal that the point of no return has been reached. In the cytoplasm, released cytochrome C binds with a protein called APOF1, apoptotic protease activating factor 1, and together they recruit a third protein, an inactive precursor of an enzyme called caspase 9. These three components, along with ATP, assemble into a large wheel-shaped molecular complex called the apoptosome. The name means, approximately, the body that initiates the apoptosome. The apoptosome activates caspase 9, which in turn activates a cascade of further caspases, specifically caspase 3 and caspase day 7, which are called executioner caspases. Caspases are proteases, enzymes that cut proteins. Once the executioner caspases are active, they cut systematically through the cell's architecture. They activate enzymes that fragment the cell's DNA into neat, regular pieces. They cause the cell's contents to be packaged into small, membrane-wrapped parcels called apoptotic bodies, which are then recognized and consumed by neighboring cells and by immune cells called macrophages. The whole process, from the initial permeabilization of the outer membrane to the final clearance of the apoptotic bodies, can be completed in a matter of hours. And throughout, the cell's contents remain enclosed. There is no spilling into the surrounding tissue, no inflammatory signal sent to the immune system. It is, in every sense of the word, a quiet death. What is remarkable about this, what deserves a moment's reflection, is how ancient and how careful this process is. Apoptosis is not a recent innovation. Its core machinery is found in organisms across the entire breadth of complex life. Fruit flies use it. Roundworms use it. Plants use a related process. The fundamental logic that a cell should be able to assess its own condition and, finding that condition beyond redemption, remove itself without causing harm to its neighbors, is something evolution arrived at very early and has preserved ever since. And the mitochondrion is the gatekeeper of that decision. The outer membrane permeabilization is the point of commitment. Before it, the cell may still be rescued. The pro-survival proteins may reassert themselves. The stress may be resolved. After it, the cascade is essentially irreversible. The cytochrome C is out. This is why mitochondrial health matters so profoundly in the context of cancer, of neurodegeneration, of aging. A cell whose mitochondria are chronically stressed, whose outer membranes are chronically closer to permeabilization than they should be, lives in a state of low-grade proximity to that threshold. And a cell whose mitochondria cannot trigger the pathway when they should, as happens in many cancer cells, which have developed mechanisms to suppress apoptosis, can persist and proliferate when it should be quietly removing itself. The mitochondrion does not merely power the cell, it holds the threshold. It is the place where the question is asked, is this cell worth maintaining? And it is, in the deepest sense, the place where the answer is given. This is not a grim thing when you consider it from a distance. The falling of leaves in autumn is not a failure of the tree. It is the tree preparing itself, clearing away what is no longer needed, making space for what comes next. A form of maintenance so ancient and so careful that it has been trusted by evolution for hundreds of millions of years. Your cells are making this assessment, quietly and continuously, in the background of everything else they do. Most of the time, the answer is continue, and so they do. Every person alive today received their mitochondria from their mother. Not from both parents, not from some mixture of maternal and paternal lines, from their mother exclusively. And their mother received her mitochondria from her mother, and her mother from her mother, and so on, in an unbroken chain stretching back through every generation of human beings that has ever lived, back through our pre-human ancestors, back through the primates, back to the very earliest mammals, and before that further and further into a past so distant it becomes almost impossible to hold in the mind. Your mitochondrial DNA is a direct, uninterrupted inheritance from a single maternal line. Every mutation it carries, every variation in its sequence, was introduced somewhere along that line and has been passed down faithfully ever since. It is a record. A long, slow, biological letter written across deep time, and you carry it in every cell of your body. Ye only the mother. The answer lies in what happens at fertilization. A sperm cell contains mitochondria. It has to. The tail of a sperm is one of the most energetically demanding structures in the human body, whipping back and forth at extraordinary speed to drive the cell forward, and that motion requires ATP, which requires mitochondria. A sperm cell packs its mitochondria tightly around the base of its tail, exactly where the energy is needed most. But when a sperm fertilizes an egg, something happens to those paternal mitochondria. They are marked for destruction. The egg cell, and this is the remarkable part, specifically recognizes the sperm's mitochondria and tags them with molecular labels that target them for mitophagy, the same cellular self-digestion process we discussed in the context of damaged mitochondria. The paternal mitochondria are engulfed and broken down within the first few days of embryonic development, before the embryo has even implanted in the uterine wall. The mechanism by which this happens has been studied in detail in several organisms and appears to involve, at least in part, the ubiquitin tagging system, the same system used to mark damaged mitochondria generally. The sperm's mitochondria arrive in the egg already tagged with ubiquitin, the molecular label that signals remove this. The egg's autophagy machinery obliges. Within days, the paternal mitochondria are gone. Why does the cell do this? The honest answer is that we are not entirely certain. One compelling hypothesis is that it prevents conflict. Mitochondrial DNA mutates at a higher rate than nuclear DNA. It lacks some of the repair mechanisms available to nuclear chromosomes, and it sits close to the electron transport chain where reactive molecules are constantly being generated. If both maternal and paternal mitochondrial genomes were present in the same cell, there could be competition between them, with consequences for the cell's function. By ensuring that only one maternal lineage is present, the cell avoids this potential conflict. Another hypothesis relates to quality control. The egg cell is, in many ways, the most carefully maintained cell in the body. It is kept in a state of arrested development for years, sometimes decades, and the mitochondria it contains have been selected and maintained with extraordinary care. The sperm's mitochondria, by contrast, have been through a demanding journey and may have accumulated damage. Removing them may simply be a matter of replacing a potentially compromised set with a carefully maintained one. Whatever the reason, the consequence is clear and profound. Because mitochondrial DNA passes only through the maternal line, it does not undergo the reshuffling, the recombination, that nuclear DNA experiences at every generation. Nuclear chromosomes from both parents are cut and rejoined during the formation of eggs and sperm, mixing genetic material in new combinations. Mitochondrial DNA simply copies itself and passes on. This means that variations in mitochondrial DNA accumulate gradually over time through mutation alone at a relatively predictable rate. This makes mitochondrial DNA an extraordinary tool for tracing ancestry. By comparing the mitochondrial DNA sequences of people from different populations around the world, geneticists can reconstruct the branching of maternal lineages across time. They can identify the point at which two populations' maternal lines diverged, and from the number of mutations that have accumulated since, estimate how long ago that divergence happened. This approach has been used to trace human migration out of Africa, to identify population bottlenecks, to follow the movement of ancient peoples across continents and islands. The deepest branches of the human mitochondrial family tree converge on a single woman, not the only woman alive at the time, but the one whose maternal line has survived, unbroken, to the present day. She lived in Africa, most estimates suggest, somewhere between 150,000 and 200,000 years ago. She is sometimes called mitochondrial Eve. Her mitochondria are in you, right now. But the story of mitochondrial inheritance has taken a new and genuinely surprising turn in recent years. In 2024, researchers at the Chinese Academy of Sciences published findings from experiments with a small roundworm called Caenorhabditis elegans, a creature just 1 millimeter long, with a body plan simple enough that every one of its roughly 1,000 cells has been individually identified and named, and which has been a workhorse of genetics research for decades. The researchers induced mitochondrial stress in these worms, disrupting the function of the electron transport chain in a way that did not kill the worms, but placed their mitochondria under significant strain. They then looked at the offspring of these stressed worms, and then the offspring of those offspring, and then the offspring of those. The effects persisted across more than 50 generations. Descendants of the stressed worms, worms that had never themselves experienced mitochondrial stress, whose parents and grandparents and great-grandparents stretching back 50 generations had also not experienced it, were measurably different from control worms. They developed more slowly, but they also lived longer. The signal was being transmitted, not through the nuclear genome, which was unchanged, not through the environment, because the descendant worms were raised under identical conditions to the controls. Something about the mitochondrial stress, some molecular memory of it, was being passed down through more than 50 generations, shaping the biology of descendants who had no direct experience of the original event. The mechanism is not yet fully understood. It may involve small RNA molecules that are passed from parent to offspring alongside the mitochondria themselves, molecules that carry information about the metabolic state of the parent and influence gene expression in the next generation. It may involve modifications to the structure of the chromatin, the packaging of DNA, that alter which genes are active without changing the DNA sequence itself. These kinds of heritable changes that do not alter the DNA sequence are called epigenetic changes, from the Greek epi, meaning above or upon. What is certain is that the finding opens a question that has not yet been answered. If mitochondrial stress in one generation can echo forward through 50 generations in a roundworm, what might be echoing forward through human generations? What might our cells be carrying quietly from the experiences of ancestors we never knew? This is not a question with an answer tonight. It is a question worth holding gently, the way you hold something fragile in the dark, carefully without gripping. The thread runs back a very long way. For most of the 20th century, the story of the mitochondrion was essentially a story about energy. Here is where ATP is made. Here is the electron transport chain. Here is the citric acid cycle. The mitochondrion was understood as a power plant, important, essential, but limited in its role. The 21st century has been steadily dismantling that picture. What has emerged in its place is something stranger and more interesting, a portrait of the mitochondrion as a deeply integrated player in cellular life, involved in signaling, sensing, defending, communicating, and doing things that nobody, even a few years ago, would have thought to look for. We will begin with light, and with a finding from 2025 that still has something of the quality of a surprise. The mammalian retina, the thin sheet of cells at the back of the eye that converts light into electrical signals, is built in a way that has puzzled biologists for a long time. In most vertebrates, including humans, the retina is, in a sense, installed backwards. The photoreceptors, the rod and cone cells that actually detect light, are not at the front of the retina, facing the incoming light. They are at the back. Light has to pass through several layers of other cells before it reaches them. This arrangement seems inefficient, and in some ways it is. But the retina has evolved a number of adaptations to compensate, and in 2025, research suggested that one of those adaptations involves the mitochondria of a specific cell type called glia, support cells that run through the full thickness of the retina, from front to back, like columns holding up a building. The mitochondria in glial cells are unusually large and unusually densely packed, and the 2025 research suggested that these mitochondria may function as microscopic lenses, structures that gather and focus incoming light, channeling it more efficiently toward the photoreceptors at the back of the retina. The physical basis for this is real. mitochondrion has a refractive index, meaning it bends light. That is different from the surrounding cytoplasm, in the same way that a glass lens bends light because its refractive index differs from that of air. A dense, spherical or ovoid mitochondrion sitting in the path of incoming light will refract that light, concentrating it. If the mitochondria are arranged appropriately, and in Muller glial cells they appear to be, they could collectively act as a distributed optical system, improving the amount of light that reaches the photoreceptors and potentially sharpening the resolution of the image formed there. This is a function that nobody associated with mitochondria. These are organelles defined by metabolism, by energy production, by the electron transport chain and the citric acid cycle. The idea that they might also be optical components, that they might be bending light in your eyes right now, quietly improving your vision, is the kind of finding that requires a certain amount of sitting with before it fully settles. It also raises an interesting possibility for medicine. If mitochondrial dysfunction in retinal cells contributes to visual deterioration, and there is already evidence that mitochondrial decline plays a role in conditions like age-related macular degeneration, then the loss of visual acuity might be partly a loss of this optical function, not just a loss of photoreceptor sensitivity. This is a new lens, so to speak, through which to view retinal disease. Now we turn to something even more unexpected, the possibility that mitochondria travel between cells. The idea that an organelle, a structure inside a cell, could leave that cell and enter another has only recently moved from hypothesis to something approaching evidence, but the evidence is accumulating. There are now multiple reports from different research groups of mitochondria being released from cells, not as a result of cell death or damage, but apparently as a deliberate secretory event. These released mitochondria, or fragments of mitochondria enclosed in small membrane vesicles, can be detected in the bloodstream and in other extracellular fluids. More intriguingly, research published in 2025 and 2026 has suggested that mitochondria released from cells in one organ can be taken up by cells in another organ, and that this uptake has functional consequences. One particularly striking line of work involves the heart. Mitochondria released from liver cells or from cells in the bloodstream appear to be taken up by cardiac muscle cells, the cells of the heart, and to induce in those cells a state of increased resilience against the kind of oxygen deprivation that occurs during a heart attack. The mechanism by which this works is not yet fully understood. It may involve the mitochondria delivering functional electron transport chain components to cells whose own machinery is compromised. It may involve the released mitochondria acting as signaling entities, triggering changes in the recipient cell's gene expression or metabolic state, rather than being incorporated as functional organelles. It may be both. The field is young and the picture is still forming. What is clear is that if mitochondria can act as messengers between organs, carrying information or functional capacity from one tissue to another, bypassing the nervous system and the hormonal system, then our understanding of how the body communicates with itself is incomplete in a fundamental way. And this leads naturally to a third unexpected discovery, the mitochondrion as a source of signaling molecules. In recent years, researchers have identified a class of small proteins called mitochondrial-derived peptides, short chains of amino acids encoded not in the nuclear genome but in the mitochondrial genome itself. The mitochondrial genome, as you may recall, encodes only 37 genes. For a long time, those 37 genes were thought to encode only 13 proteins and the RNA molecules needed to make them. But hidden within some of those sequences, readable in a different frame, or from regions previously thought to be non-coding, are sequences that encode small peptides with their own biological activities. One of the best studied of these is a peptide called MOTC-C mitochondrial open reading frame of the 12S ribosomal RNA type C, if you want the full name, though the short name serves perfectly well. MOTSC, was identified in 2015, and since then it has been found to do something remarkable. It rises sharply in the bloodstream in response to exercise. It circulates, it enters cells, and inside cells it influences metabolic processes in ways that partially mimic the effects of physical activity, improving insulin sensitivity, supporting the use of glucose and fatty acids as fuel, protecting against metabolic deterioration. Researchers are now studying MOTSC as a potential therapeutic molecule, a way to capture some of the metabolic benefits of exercise in a form that could be administered to people who cannot exercise, such as those with severe illness or disability. The work is still at an early stage, and many questions remain about how MOTSC behaves in humans across different ages, conditions, and contexts. But the broader point is striking. The mitochondrion, this ancient bacterial guest, this ATP-producing organelle, is also a hormone-like signaling system. It produces molecules that enter the bloodstream and act on distant tissues. It is not just a power plant, it is a gland, a sensor, a communicator. The story of the mitochondrion keeps expanding. Every decade, it turns out to be involved in something that nobody expected. And the sense you get, following this research, is not that we are converging on a final picture, but that we are still in the early stages of understanding what this ancient organism, living inside us, generation after generation, is actually doing. It is patient. It has been here a very long time. It is in no hurry to reveal everything at once. You are tired. Perhaps you have been tired for a while, not just tonight, but in a deeper way, the kind of tiredness that accumulates across days and weeks, the kind that sleep promises to address, but sometimes seems only to touch the surface of. If that is where you are tonight, this chapter is especially for you, because it turns out that the tiredness you feel, that specific, heavy, insistent pressure towards sleep, is not simply a chemical signal in your brain. It is, in a very literal sense, a message from your mitochondria. In 2005, a team at the University of Oxford, led by Professor Jérôme Miesenbuch and Dr. Raffaele Sarnataro, published a paper in the journal Nature that described, for the first time in careful mechanistic detail, how sleep pressure builds at the cellular level. The study was conducted in fruit flies, Drosophila, the small insects that have been one of biology's most productive research subjects for over a century, but the mechanisms it uncovered are deeply conserved across evolution, meaning they are likely to operate in broadly similar ways in the neurons of mammals, including humans. The researchers focused on a cluster of neurons in the fly brain called the dorsal fan-shaped body neurons. These are sleep-regulating neurons, cells whose activity is directly linked to whether the fly sleeps or stays awake. When these neurons are active, the fly sleeps. When they are suppressed, it stays awake. They are, in a sense, the fly's sleep switch. What Miesenbuch and Sarnataro's team found was this: as a fly stays awake, the mitochondria in these specific neurons gradually become overloaded. The electron transport chain is working hard, processing the high metabolic demand of a waking, active brain. And as the load increases, the chain becomes less efficient. Electrons begin to escape before they complete their journey to oxygen at complex 4. These escaped electrons, single electrons, unpaired, energetically unstable, react with oxygen molecules to produce what are called reactive oxygen species. A reactive oxygen species is a molecule that contains oxygen and carries an unpaired electron. The unpaired electron makes it chemically reactive. It wants to bond with something, and it will do so rapidly and indiscriminately, damaging whatever nearby molecule it encounters: proteins, lipids, DNA. The most common reactive oxygen species produced in this context is a molecule called superoxide, formed when a single electron reacts with an oxygen molecule. Superoxide is rapidly converted by an enzyme called superoxide dismutase into hydrogen peroxide, which can then generate other reactive species. In small amounts, reactive oxygen species are normal byproducts of mitochondrial metabolism, and the cell has antioxidant systems specifically designed to neutralize them. But as wakefulness extends and the electron transport chain in the sleep-regulating neurons becomes increasingly overloaded, the rate of electron leakage increases. The reactive oxygen species accumulate faster than the antioxidant species accumulate faster than the antioxidant systems can clear them. And this accumulation, this building oxidative load within the mitochondria of those specific neurons, is the signal. The neurons sense the reactive oxygen species. The sensing triggers changes in the electrical activity of the neurons. They become more likely to fire in the patterns associated with sleep. The more the mitochondria leak, the stronger the signal, and the stronger the pressure to sleep. This is a beautiful and somewhat moving piece of biology. Sleep pressure, that feeling of heaviness you know well, that specific weight behind the eyes, that pull toward unconsciousness, is, at its cellular root, a distress signal from overloaded mitochondria. And the leak is the message. Enough. Rest now. The Oxford team went further. They showed that by artificially manipulating the balance between mitochondrial fusion and fission in these neurons, they could change how long the fly slept. When fission was promoted, when the mitochondrial networks were pushed toward fragmentation, the fly's sleep was reduced. When fusion was promoted, when the networks were encouraged to merge and reform, the flies slept more. The mitochondrial architecture was not just a passive reflection of the sleep state, it was actively participating in its regulation. This connects everything we have covered about mitochondrial dynamics to the specific, practical reality of sleep. The shift toward fragmentation during wakefulness and the shift back toward fusion during sleep are not incidental. They are part of the mechanism. So what happens during sleep? What do the mitochondria do when the pressure is finally released? The answer is that they rest, and in resting, they repair. During the deepest stage of sleep, the stage characterized by slow electrical waves moving across the brain, slow wave sleep, sometimes called deep sleep, the metabolic demands on neurons drop substantially. The oxidative load that accumulated during wakefulness is gradually cleared. At the same time, mitophagy, the targeted removal of damaged mitochondria, is promoted during sleep. The mitochondria that were most damaged during wakefulness, those whose membrane potential dropped most severely, those that accumulated the most oxidative damage, are preferentially tagged and removed. Their components are recycled. New mitochondria are generated to replace them in a process called mitochondrial biogenesis, which is also upregulated during sleep and in the hours following it. And during the deepest stages of slow-wave sleep, the pituitary gland releases growth hormone, a pulse of it, the largest pulse of the day. Growth hormone is known to support cellular repair and renewal broadly, and there is evidence that it specifically promotes mitochondrial biogenesis and the repair of mitochondrial membranes. The deepest sleep and the most powerful growth hormone pulse coincide, and together they create the conditions in which the cell's mitochondria can be most effectively renewed. The mitochondrial networks, fragmented by the demands of wakefulness, begin to fuse. Long, interconnected structures reform through the matrix of the neurons. The resources that were isolated in small, separated units during the stressful hours of the day are shared again, pooled again, averaged out across the network. Damaged sections that could not be immediately cleared by mitophagy are diluted by fusion with healthier partners, their dysfunction buffered by the health of the broader network. It is, in miniature, exactly what sleep feels like from the outside, a restoration. A reforming of what was scattered, a return to something more coherent and more capable than what existed at the end of a long day. Consider what this means for the nights when sleep does not come easily. When you lie here, in this quiet, and the mind will not slow, and the body does not release, the mitochondria in your sleep-regulating neurons are still sending their signal. The reactive oxygen species are still accumulating. The pressure is real. It is physical. It is cellular. It is not imagined. The body wants to sleep. The ancient engines are asking for it. Whatever is keeping you awake, the thoughts that will not settle, the anxiety that finds new shapes in the dark, is working against something very old and very insistent, which is perhaps a reason to be gentle with yourself about it. The difficulty of sleep, when it comes, is not a failure of will. It is a gap between what the cells need and what the mind will allow. And the cells are patient. They will wait. They have been waiting, in one form or another, for 2 billion years. And here, in this moment, wherever you are, whatever you are carrying from today, the day is behind you now. Its demands have been made, and most of them have been met, and the ones that haven't will still be there tomorrow, and tomorrow is a long way off. Right now there is only this, the quiet and the dark, and somewhere inside you, countless small ancient engines beginning to slow. They are not failing, they are resting. There is a difference, and it matters. The mitochondria in your neurons have been leaking their quiet signal for hours, building the pressure that brought you here, to this bed, to this stillness. The signal has been heard. The response is already beginning, in the electrical patterns of your brain, in the shift from wakefulness toward the first soft stages of sleep, in the gradual lowering of metabolic demand that allows the reactive oxygen species to clear and the antioxidant systems to recover and the damaged sections of the network to be quietly removed and replaced. You don't need to do anything with any of this. You don't need to remember it. The cells already know it. They have always known it. This is not knowledge that needs to live in the conscious mind. It lives in the membrane, in the protein, in the ancient circular molecule of DNA that has been copied and passed down through every mother and grandmother and great-grandmother back to the very beginning of complex life on this planet. The mitochondrion that is slowing down right now in a neuron somewhere in your brain is a descendant of a free-living bacterium that swam in a warm sea 2 billion years ago. It has survived everything, every extinction, every ice age, every catastrophe that has reshaped this planet. It survived by being useful, by knowing, in the only way a molecule can know anything, when to work and when to rest. It knows now. The networks are beginning to fuse. The long threads are reaching toward each other in the dark of the cell, membrane flowing into membrane, matrix mingling with matrix. The damaged and the worn are being quietly gathered and removed. The new is already forming. Somewhere behind you, the day is growing very distant. The thoughts that felt urgent an hour ago are losing their edges, softening into something less defined. The body is heavy in the way that is not burden but release. The weight of muscles that no longer need to hold anything up. The weight of a mind that no longer needs to solve anything. The ancient engines are quieting. And so, gently, can you sleep well.